this is a cover lesson just to go over all of our heredity notes so far, because I think a few people are still struggling to just grasp what gene interactions are. So if we go back to the beginning, what I originally showed you was that Mendel looked at gene interactions, but he looked at gene interactions between genes. I'm just going to make this bigger here. Genes that were not interacting with each other. They didn't affect each other. So the color of a seed's coat or the shape of a seed, they didn't affect each other. They are independent of each other, and that's a really important thing to note here. That independence means that whatever happens with the seed shape doesn't affect what seed color it gets. It's dependent on the allele that they inherit. Now, when you look at these two different traits in a single Punnett square, we have some patterns that come out. Now, always remember, Mendel always started with true breeding plants, and then he went and he crossed them. So true breeding means they only had one allele. He crossed them, and when he crossed them, that was his F1 cross. When he crossed them, he realized that all of those plants came out with 100% the same characteristic. And that was a pattern that was always the same. And that same characteristic, or that same trait, was the one that was dominant. Mendel called that the dominant trait. Then if he took that F1 generation and he crossed them again, the F1 cross gave out a new pattern. So the kids actually showed that the recipe for what trait was hidden was still there, it just had been masked. And that's how Mendel came up with the dominant and recessive ideas. So when we have this F1 cross, first of all, because they came from true breeding parents, all of the animals in that F1 generation had the same trait because they were all heterozygous. So in the case of the true breeding plant for color, they had a dominant gene for yellow and a recessive gene for yellow. Okay? And in the case of their shape, they had a dominant gene for round and a recessive gene for wrinkled. And of course, in the F1 generation, if all of them had those heterozygous genotypes, that meant that they were all, in this case, yellow and round. But that didn't mean they didn't have the recipe for wrinkled or green, but they were hidden. And you didn't see that until you crossed that F1 generation with itself and ended up seeing what the F2 generation had for us. And that's what these crosses are. An F1 cross, the heterozygous yellow and the heterozygous round, and you end up with a new pattern that came out in that next generation, which was 3 yellow to 1 green, because you ended up with a homozygous yellow, a homozygous round, and 2 heterozygous yellow ones for the color, and the same sort of one over with the shape. You had a homozygous round, homozygous wrinkled, and then heterozygous round, which of course ended up being wrinkled anyway because the round takes over. And you end up with that 3-to-1 phenotype pattern. Now that 3-to-1 phenotype pattern is the thing you've got to remember. That is a very definite pattern. 3-to-1 is what we see when we have that F2 generation breed. Okay? Now, we can look at these things separately on separate Punnett squares, but in reality, we've got to look at these things combined. So what they then started to do was they took these traits and they combined them on a Punnett square called the dihybrid cross. Now down here is that dihybrid cross. I've written it all out. I'll just make that a little smaller. Yep. This is that same 2 Punnett squares but put on a dihybrid cross. So in this case, instead of just having one allele up here, we had to have 2, whether they had yellow or round or wrinkled and green. So on this is exactly the same as above. On our F1 cross, we had seeds here, which were heterozygous yellow for colour and heterozygous for round. See, they were both crossed together, and when you look at the alleles that they can have, that means they can only pass on one allele for colour and one allele for shape. These are the 4 possible allele combinations that they could come up with, and that's what I've put on my dihybrid crossover here. Okay, so they could have passed on the dominant for yellow for colour and the dominant for shape, which is round, or they could have had a dominant and then a recessive that they passed on, either here or here, or they could have had 2 recessive genes passed on, the green and the wrinkled. Cross them, and this is exactly what you get here. My F2 generation, again, there's the genotypes, and I've drawn in the phenotypes, and they again come out with this lovely little mathematical pattern, 9 to 3 to 3 to 1. That's the mathematical pattern you get when you have 2 traits on a dihybrid cross that do not interact with each other. Okay? When they do not interact, when they don't affect each other, you always end up getting that mathematical pattern there, 9 to 3 to 3 to 1. That's evidence when you have 2 generations, so your P1 generation, they breed, they make your F1 generation, you cross your F1 generation and make another generation F2. If they come out in a 9 to 3 to 3 to 1 ratio, that means that those genes don't affect each other. That's your evidence for that. Okay, so we can actually figure out from looking at the ratios of what's bred whether or not the genes affect each other. Now, what happened was they started doing these crosses, and all of a sudden some of these patterns started to disappear. And it was then that they realized that some genes do have the ability to affect whether or not another gene is actually present or not. And we find out if those interactions are happening by looking again at this F2 generation ratio. Oops, just undid that. My F2 generation ratio is really, really important. So let me show you a few. So there's several gene interactions that we're going to be going over, but remember, first of all, there's just 2 genes in play here. Okay, one gene usually affects whether or not another gene gets to show its trait. So the first one we went over is this supplementary one. Now this is where one gene actually controls the first step of whether or not another gene is shown, and because of that, if the first gene is not present or doesn't make its protein properly, the other one can't work at all. So think of it sort of like a control relationship, where the first gene actually controls whether or not the second gene can actually show itself or not. Okay, because it controls the steps. That's called supplementary. Then you have a relationship where the gene actually specifically inhibits another gene from actually showing itself. Okay, now this is called an inhibitor. So its job is to actually stop another gene from making a protein. So if it's got any dominant traits showing, then the other one won't show at all. So that's the inhibitor relationship. Then we went and looked at complementary. This is where you need both genes come together and both of them have the steps in the process to make a trait. Both of them have to contribute part of the recipe, and without both of them present, you get no trait at all. Okay, that's called complementary. And the last one, which is a little bit different, which is called collaboration, which is where both genes have their own trait that they show, but when you mix them, it comes up with an entirely new trait altogether. That's called collaboration. So let's quickly go through each one of these and think about how it affects that ratio, because that ratio is what you're going to be having to use if you really want to show what you know here. So here's supplementary. So let's have a look at the supplementary relationship. Now, supplementary is all about A is a gene that controls whether or not B can actually show itself or not. If you don't have A making a protein, in other words, there's a big A gene there, okay, a dominant one. If A doesn't have a dominant gene there, then B won't work no matter what. So that's supplementary. So if you have the homozygous or the heterozygous version of A, B can show. If you have the homozygous recessive version of A, the protein doesn't work, and therefore B can't show itself, even if there is a big B present. So think of it in the case of dogs and mice. Both of them have this thing going on with their coat colour. Brown is little b, the recessive version, and black is either big B, or big B, little b for pigment. So if they have those pigments there, they have the dominant version of the B gene there, then they will get black. If they only have the recessives, they'll be brown. But they don't have the ability to put down the pigment and to actually get it into the coat without gene A. So if they don't have gene A, they end up having a yellow coat. So if they have two little A's here, doesn't matter what they have for their B protein, they'll end up being yellow. Okay? Now, in the case of these mice, all they need is one gene A at least in order to put out the colour. Now, to find all this out, what they did was they found pure breeding albinos and pure breeding coloured animals and started interbreeding them to go through the P1, F1, F2 generation, just like I showed you before. Remember, the F1 generation is always going to be a heterozygous version of your traits. So here's my F1 generation. It's a heterozygous A, heterozygous B crossed together. Now let's look at what traits are going to come out, remembering that A controls B. You can only have a brown or a black coat if A actually has a dominant allele. So you're going to have colour here, you're going to have colour here, you're going to have colour here because there's an A here, here, all the way down here. You're going to get some sort of colour because A is actually present. But not in these guys here. Notice here there's double A, little a. That means there's no colour. These guys are going to end up being yellow or white if they're mice. Yellow-white, because they end up not producing any colour at all. Regardless of what B genes they have, A gene isn't there to allow the colour to occur. So let's have a look at this pattern a little closer. Okay, when you have a look at their phenotypes, anything that has a dominant A is going to have colour in it, like I said. If it's got one or two dominant Bs, it's going to be black because it allows for the black gene. If it's the recessive little b, it ends up brown. And we end up with a new phenotype ratio here. We have 4, 5, 6, 7, 8, 9 to 3 to 4. We end up with a new phenotypic ratio, which is unique to supplementary relationships. Okay, so when you have a supplementary relationship, you have 9 to 3 to 4, and 4 is the recessive version, in this case white or yellow. Okay, so if you see on your F2 generation any 9 to 3 to 4 ratios in your phenotypes, then you know it's actually a supplementary relationship because A is controlling whether or not B gets expressed. In this case, A, which makes a protein to allow B to make the coat colour. If A is not present, B can't make the coat colour. Okay? So that 9 to 3, 4 to 4 relationship is unique to supplementary. Let's look at another one. Okay, so the next one is inhibitor relationship. We also call these a recessive epistatic relationship because B only gets to show when A is in its recessive form and doesn't work. Okay, so we sometimes call them recessive, but just call it inhibitor. That's a lot easier to remember. B only gets shown if A doesn't work. If A does work, in other words, there's any dominant A's or big A's, then B is suppressed and doesn't get to show. Now, the example we showed you in this case was the zucchinis, where the coat color, well, not coat color in this case, the color of the fruit is actually determined by the B gene, and the color of the fruit is either, if there's a dominant relationship which gives a yellow color, so big B, or big B, little b, in this case will give yellow, or if you have just the recessive gene, you'll end up with a green fruit, okay? Now remember, in these zucchinis, the letters are slightly different. I'm just trying to get you guys to realize that A is the gene that controls, and B is the one that's trying to show its trait, but it's affected by A. The letters will be different when you actually go in and do all of your different types of practices. But you have to get in your head which gene is affecting which. In this case, in our practices, A always affects B. But in this case, A is trying to stomp B down and not let B have a go. So you can have these green and yellow versions of your alleles, but if A has any dominant letters, it will make a protein that stops B from even showing itself. So let's have a look at our little dihybrid crossover here. Again, we started with purebred zucchinis up the top, and then what we did was we've then gone and got a F1 generation which is heterozygous, big A, little b, little b, big B, little b, crossed that F1 generation, and this is my F2 generation. We're looking for the pattern. So we'd always look for the pattern from the heterozygous cross of the F1 generation to see if it gives us a hint as to what type of relationship it is. So firstly, anything that has A is not going to show any trait whatsoever, or big A, because big A suppresses any B's. So these guys here, oops, yellow is a really bad color to use here, just a minute. Just give me a second. Now I'm using grey here, but it's supposed to be white. Anytime you have a big A present, you're going to have no color coming forth because your B gene is completely stomped on. It can't do anything. So all of these guys end up being white. The only time you're going to get color is down here where your A is recessive. Now remember, big B, or big B, little b, the dominant color is yellow. So these guys here are going to be yellow, and this one here is going to be green. Notice we have a new ratio that has just come up. So when you have an inhibitor gene like this, you have another unique ratio. The ratio is 4, 8, 9, 10, 11, 12, 2, 3, 2, 1. Okay, and that is a unique relationship too. Okay, that indicates that there's an inhibitory action happening. Okay, now in complementary genes, this is where both of them actually do some of the steps to get the job done for a phenotype. So in the case of purple pea plant flower color, both A and B make part of the process for getting the purple color in the flower, and if either of them don't work, you end up with a white flower with no color. So in this case, we actually need both of them to have at least one dominant allele in order to get color. So let's have a look at the relationship or the pattern that we see with this now. So again, here is my heterozygous cross of my F1 generation. Put them up here with all their alleles. My F2 generation is going to have a specific pattern in it that shows me that this is definitely a complementary relationship. So just a minute. So in this case, any of the combinations where you do have at least one dominant in both alleles, you're going to get purple, because the A and the B gene have both got a job to do in order to get the job done. But look here, we've got yet another unique sort of pattern here. So in the case of complementary relationships, number one, you only get two phenotypes. One where the gene products are working together to make the end product, and one where they don't. So you drop down to two phenotypes, and there's another definite ratio, 9 of where they work to 7. So as soon as you see these sort of ratios happening here, and also note that there are only two phenotypes, that's when you start to see that, oh, with that sort of ratio there, if you get that from an F1 cross, your F2 generation has a ratio of 9:7, that's how you determine that it's a complementary relationship. So see, with each one of these, we've deviated from what was that original 12-3-3-1 relationship that I showed you before. Now, the last one that I was also going to go into was collaboration. And this is not where one gene suppresses another or allows another to show, but these are where you have two genes that have their own distinct trait, but when you mix them, you end up getting a blend of the two characteristics. So it's sort of like incomplete dominance, but with two genes, not one. Okay? So let's just have a look at it. Here we have the roosters with the combs. If you have a single comb, you don't have either the R or the P gene, or in my example here, A or B. But if you have just the P gene, you end up getting a P cone, and that doesn't matter whether you're homozygous or heterozygous for P. You just have to have one dominant allele. If you have one dominant allele for the R gene, you get a rose comb. But if you mix the dominant alleles, the rose and the P, you end up getting a completely different gene phenotype here called the walnut. So let's just go back to our example here and show you how it affects the ratios. Okay, so again we've got a heterozygous blend of the A and the B, or in the case of the chooks, the P and the rose combs. So if you've got heterozygous ones of those, you're going to end up with that walnut trait, because both of them are going to show and turn it into that walnut trait. Now, I'm looking for first my A gene, which is going to be my P. Now, if I have any dominant A's, that means it's going to show, but not if I have a dominant B as well. Only A's where the B is not showing at all. So here, and here, here. And I think that's all of them. Now, that is where the P gene here will actually show through, but there's no B ones to mix with it because the recessive version doesn't work. Now let's look for the other version, the rose comb. So let's get a nice rose color here. So this is where you have no dominant A's and only dominant B's. So here, and here, and here, and that looks like all of them. So they're the only times with this cross that you'll see just a P gene or just a rose gene. The rest of them you have both a dominant A and a dominant B, and they're going to combine to give a new phenotype, remember? If you have both the A and the B gene in its dominant form, you get this other phenotype called the walnut, which is a mixture of both the dominant genes. So let's mark them out. And that's going to end up being all of these guys up here. I've given it a brown color because it's, you might have noticed that walnut comb doesn't look anything like the original two phenotypes. Now last but not least, you've got this little recessive guy down here which actually has none. And that means that he doesn't show either a peanut comb or the rose comb or the combined version of the two. So again we have one unique sort of phenotype ratio coming out here. So 1, 2, 3, 4, 5, 6, 7, 8, 9 to 3 to 3 to 1 again happening. So when you have this collaboration gene, you end up getting back the same sort of ratio that you had when you had your independent alleles before. Okay, the original ones like the yellow coloured P and the wrinkled P. The only difference here is that this one here is a new phenotype that has not been seen before. It's a mixture of the two. Although you have the same type of ratio, you have a new phenotype emerging that wasn't there before. Okay, and only one of the recessive version. Now in the case of polygenes, they actually break the rules. This is not where you have one inhibiting another or one allowing another to be expressed and stuff. This is actually where both of them end up being blended versions of each other, depending on how many and what type of alleles you have. So you end up with a continuous scale, and you end up with more than 14 phenotypes, just like with human skin color. This is a very simplistic version of what human skin color would be like if it was only controlled by 2 alleles or 2 genes, but it's actually controlled by more. And even with each one of these different combinations, you get quite a few different colored skin types, and as you know, there's even more than that. So polygenes is where you end up with a lot more than 4 phenotypes, and there's a continuous scale going from one extreme to another. So they're quite easy to pick out because you don't have that definite ratio for the phenotypes. They actually break the box and just spread themselves out and have this continuous scale of different types of phenotypes that you see.